It'll be by Yasir Gallero-Salas. He's from a company called Cupra, based in Denmark. He will talk about their pipeline for high-throughput and quantitative whole brain light sheet imaging. Looking forward to it. Thanks.
Thank you for the nice intro and the opportunity. I think it's great to have such a big audience to listen to our talk. So as I said, she said I'm a scientist at Cupra, and we are a biotech that, among other things, we specialize in applying light sheet microscopy in clear samples in the preclinical drug discovery. So I will first give you a very brief introduction of the light sheet process that we use. Then I will talk about pipeline, divided in first preserving endogenous fluorescence, typically used in biodistribution studies of drugs. And then I will give you a second pipeline of how we can label samples in 3D with immunohistochemistry, in particular neural activity and cell type markers. Finally, I will briefly mention how we deal with this data, how do we align it to a reference atlas. And finally, I will talk to you about our Neuropedia, our open platform to share digital maps of the brain. So typically, we are working with clear samples that are labeled, and we typically use two strategies. Either we inject in vivo a compound or drug that is fluorescently labeled, or we use the iDisco technique that uses primary and secondary antibodies to label our sample, which in this case, we are focusing on the brain, but we also are applying to other organs, like the kidneys and the lungs. After labeling, there is a final step of tissue clearing by getting rid of the lipids and matching the refractive index of the organ with different chemicals. And after that, we applied light sheet microscopy. So as it's been introduced, the light sheet microscopy is characterized by an objective that shapes the light into a sheet, and so that we can use this sheet to scan in a sample by sample and section by section in the whole Z direction of the organ. So per section, we will find an image such like this, where autofluorescence is shown in grayscale, and the blood vessels are labeled in green. However, from a single section, it's very difficult to visualize the whole structure of the blood vessels. But once you start, stacking all those sections, you can clearly visualize the whole vasculature in 3D. This is one of the most beautiful examples of how 3D microscopy overcomes some of the limitations of more classical 2D histology. And so, in order to have a high-capacity 3D imaging platform, we think that we need to have three pillars that need to be solid and communicating to each other. And the first one is the lab. We need to have an automatic pipeline, in this case, a robot that can follow the whole iDisco platform, meaning that it will take a couple of hundred brains, and they will label them and render them clear. Then the next pillar would be the microscopy. And so we are working towards having a fully automated microscope that can run 24 hours imaging brain samples. We also require a third pillar, which is the data analysis, which is very important. So for that, we have a semi-automatic pipeline that can produce quantifiable endpoints, such as signal volumes or cell counts. We typically image each brain per channel in 45 minutes, and we typically work on the order of hundreds of brain per month. So first, I will mention about how we do studies where the goal is to preserve endogenous fluorescence. So typically, these studies have an in vivo phase in where we inject a fluorescently labeled drug or compound, and we compare the biodistribution of it versus a vehicle. After that, we render the brain clear. We avoid any photobleaching, any type of bleaching to preserve the signal injected in vivo. And after the organ is clear, we typically use DB Then we run it through the microscope, we perform the image analysis, and the result is a quantifiable signal intensity or volume per brain area. And one of our limitations in this study is that we normally work with very dim signals. For example, when we inject Excitin-4 labeled with sci-fi, in vivo, we see some signal in the hypothalamus, which is typically very dim. So we are trying different clearing reagents to see if we can improve not only the signal intensity the day after clearing, but also across time. And so in this example, we are comparing a DBE, which is classically used as a clearing agent, versus ECI, a non-toxic clearing agent. And we can see that both at day one after clearing, here in a zoom-in of the hypothalamus, the signal is much higher in the brains clear with ECI as compared to the ones with DBE. But not only that, it seems that the ECI also preserves the signal much better across time. If you look at the literature, you will see that DBE contains some peroxides and aldehydes that might kill your fluorescence. In our second pipeline, we label markers of neural activity and cell types in 3D. In this case, we typically compare the influence of drug versus the vehicle in a given marker. For instance, CFOS, which is a neural activity marker. And in this case, we do have to label this marker, and for that, we typically use primary and secondary antibodies, although we're also starting to explore other options like conjugated antibodies. And after that, it follows a similar pipeline of imaging, reference to an atlas, and signal quantification, typically cell numbers per brain area. And so we are applying successfully this pipeline to different antigens. For instance, I'm showing you a staining of TH in mouse, where we can have cellular resolution across the whole volume. And another example of this type of study where we label CFOS, as I mentioned, an activity marker. So that brain, for instance, was recorded within 1.5 hours. So it's 2 channels of the fluorescence in grayscale, and the CFOS is in glow scale. So we have enough resolution to image and quantify single cells that are expressing the CFOS. We have started also applying this process to more challenging samples as the rat brain, which is bigger and have a different tissue density. And it has some challenges when it comes to clearing, but here I can show you an example of a blood vessel labeling in a rat. And as a proof of a principled study, we treated mice with 6 different appetite regulating drugs. Here's one example of how the raw data looks in 3D. And we analyzed the CFOS print of each individual drugs. We can see the average CFOS response across the brain, which we can later on put together in a reference map and see that areas in white, they seem to be participating more often in these appetite regulating drugs. So in order to do that, we have to have our data analysis pipeline, which aligns each brain into an average maps in order to finally visualize an average drug response. And for that, we have to align each brain to a common template who has an annotation of the brain atlas. So we typically use the annotation from the Allen Institute. And then tomorrow, on Friday, Johanna, another scientist at Gubra, she will tell you more about how we do this process and why we need a iDisco-specific reference map. The final result is that we can either extract signal intensity or cell number per brain regions. In this case, more of 250 brain regions. Finally, I will talk to you about Neuropedia, what is Neuropedia, and why should we care about it. So Neuropedia is our open access database for sharing digital brain maps. We always confronted of the lack of information to take decisions, in particular to preclinical drug discovery, but also in academic life. When we find CFOS activity in a given nucleus of the brain, this is the only dimension that we know about this, but what else is in this nucleus? What type of markers, what type of peptides, what is the connectivity of that nucleus? What is the anatomy? So for that, we thought that given our capacities of imaging and data analysis and references to an atlas, we are in a good position to collaborate with labs across the world. And for that, we set a collaboration where we can receive 3 brains from any lab that is interested. We will clear them, image, and produce a reference map that we will share back with you, but also with the community. And the final result is that we can overlay different information in a single map. For instance, in here we can see that we start with a CFOS response of glucagon-like peptide, which is a peptide that can be injected and produces this CFOS fingerprint. But also now if we overlay this map with the expression of GLP-1 receptor and at the same time also overlay it with the biodistribution of GLP-1 and the anatomy, where are these leaky capillaries, and finally the cell markers, what type of neurons are there. So we think that by combining and joining forces, we can have a better understanding of what's going on in a particular brain area, and then we can take more informed decisions. Thank you so much for your attention. I will have later in the night a poster with the similar results, and please come talk to me, and I'm really happy to talk about anything in light chain microscopy or drug discovery. And I will happily take questions.
That looked really good. Thank you. Questions from the audience, please. There, over. Go ahead.
So if I understand well, this is a company, right? Google.
Yes.
Yeah. So is that your business model? Is that you want to actually do the processing of the sample and the analysis? I'm just curious about the company side of all this.
We typically work for other biotech, pharma, universities. We run in our department we run the studies for 3D imaging. The Neuropedia, I think that's what your question refers to, is an initiative which stems from our company, but the goal is to have an open access platform, so that if you run a lab and you have an interest in anatomy study, or injection, or transgenic animal, and then you can just send us 3 brains, and then we do the clearing, imaging, analysis. We give back to you the videos, and then we post on the webpage of Neuropedia, which everybody can access. And there you can do the overlay. You can get a better information of what's going on in a particular brain area.
More questions? Over there.
Great talk here, Alice. Thanks. Regarding Neuropedia, are you interested only in mouse brain or also zebrafish brain, where you could save the clearing step, for example?
So for the moment, we are starting to focus on mouse brain, which is one of the models widely used in drug discovery. And that's an avenue to explore. I think there's more and more interest in rats and other model organism, but at the moment we focus on the mouse brain.
Okay, thank you very much for the questions. Will be interesting to see how the transgenic models look like with your system.
Thanks a lot.
Thanks a lot.